So viewers, this is my second lecture on animal physiology. Today I will discuss about the hemoglobin estimation. First of all, we have to know that what is hemoglobin? Hemoglobin is a heme protein pigment that is found in the erythrocyte of mammalian blood. It is composed of mainly two parts, globin part and heme part. Globin part is apoprotein portion, and it is tetrametric configuration. It is constituted with paired peptides like alpha, beta, gamma, delta, and each of these peptide bond is attached with the heme protein. Heme is a prosthetic group and it is termed as ferroprotoporphyrin 9. It is constituted with the 4 pyrrole group. Look here, the pyrrole groups A, B, C, D. They are connected with each other with 4 methylene bridge. AB pyrrole has 2 binding sites, that is methyl and phenyl. Like that, C and D pyrrole group has also methyl and propionate site. But peculiarity is that they are, that is C and D are arranged in reverse order. Secondly, the character is, look, that protoporphyrinogen oxidizes it and transform this structure 3 structure to 9th structure by the oxidation of these methylene bridges. Secondly, ferrochelatase utilizes the Fe of transferrin to incorporate Fe2 plus into the center of this spiral ring. This iron component has, this iron molecule has 6 coordinating bond. 6 out of that 4 coordinating bond, that is this one, this one, this one and this one, remains in the non-axial plane, bounded together with nitrogen molecules of 4 pyrrole bands. 1, 2, 3, 4. Another axial bond that is in the another plane has this one axial bond, which 5th bond is bounded with the same peptide bond structure of globin structure at the 8th position of F alpha helix. Another 6th bond, 6th hand, binds with oxygen. It, when binds with oxygen, it is termed OXYHAEMOGLOBIN. It may be bind with carbon dioxide or carbon monoxide. Then it is termed as like that, that is hemoglobin, etc., etc. But this type, when oxygen binds here, this is the structure of OXYHAEMOGLOBIN. Now look, it is termed there now the ferroprotoporphyrin group that gives the coloration. The color is red. The red pigmentation is found in RBC of the blood. Our it is the estimate this term amount of pigment, that is heme protein, and to do that we have to enter into the RBC cell of the blood. How can I do that? Before we estimate the hemoglobin proteins, we have to know some standard values, and like this: 1 deciliter, each deciliter contain 15 gram of hemoglobin. Each red cells contain 20 picogram. Each unit of corpuscular concentration contain 35% hemoglobin. If the patient become anemic, then it goes down up to 20%. Each male species, that is male human, contains in each deciliter and 14 to 17.5 gram. In case of female, it can per deciliter it counts 11.5 to 16 gram. But it is to be remembered that determination of MCH, MCHC, or even the complete blood cell CBC, hemoglobin per liter or hemoglobin per deciliter is primarily required. So our target is to determine the hemoglobin per deciliter or milliliter. How can we do that? In the laboratory, college laboratory, we do it with SAHLI'S hemoglobinometer, because SAHLI is this hemoglobinometer is easily available in the market. It is economic, it is portable, but most important is that it does not require any pre-calibration. Moreover, the enumeration has been but can be determined directly from the from its instrument. Now, how this functions? This instrument is for the acidification of hydrochloric acid. Hydrochloric is acidified. Blood is acidified with hydrochloric acid. By this time, hemoglobin transformed into acid hematine hydrochloride. Color is developed from red to brown. Then the brown color is compared in the comparator box of the hemoglobinometer. Amount of the hemoglobin present there then be counted in the from the experimental tube directly in gram or percentage value. Now we should know that how the reaction is done during this reaction with hemoglobin and hydrochloric acid. Hydrochloric acid is taken in 0.1 normal solution. It is hypotonic in nature. If we use hypertonic solution, if we use isotonic solution, or we use the concentrated hydrochloric acid, or other than hydrochloric acid the alkali medium, the results will not be up to the mark, because hemoglobin structure will be reacted otherwise and will constitute a different type of molecules, hemoglobin molecules. Our target is to hemolyze the RBC to exclude the heme molecules from the cell sap. Hemoglobin does this function. Hydrochloric acid does this function. Hydrochloric acid dissociate the heme part and the globin part. From the hemoglobin structure, this part is globin part, and after dissociation, that ferroheme structure is formed here. This reaction between hemoglobin and hydrochloric acid occurs in 1:1 stoichiochromatic reaction. For this reason, in the second reaction, hydrochloric acid and the ferroheme occurs in equimolar proportion. Look here, 2 moles of 2 atoms of ferroheme and the hydrochloric acid react with each other. During this reaction, hydrochloric acid dissociated into hydrogen, reacts with oxygen and goes out as H2O, but chlorine is incorporated and reacted with iron, make a ionic bondage with it and form a chloride structure. Fe2 then becomes Fe3. Ferrohemi become ferrohemi. Color become brown. Okay. Now this is the reaction portion. Now next we want to go or see how it happens. Phenomenon happens in the SAHLI'S hemocytometer. Hemocytometer has a comparator. Comparator box is used for matching the color or developed color, that is red to brown. There is diluting tube which is graduated and estimated result is found directly from it. A capillary tube is there to draw the blood sample from the human body. Glass rod is there for mixing between the HCl and blood. A dropper is used to add the distilled water for making dilution again. Distilled water is used making dilution. Alcohol, 90% approximately, is used for sterilization. Hydrochloric acid, N/10 hydrochloric acid is required for this reaction, that is used for hemolysis of the RBC. Now we are going to explain how this N/10 hydrochloric acid can be prepared. You know, the hydrochloric acid has the molecular weight 36.5, that is 35.5 plus 1, that is 35.5. Then 3.65 gram is required to make it 1 liter by distilled water. By that means you get 0.19 normal solution. But the availability of hydrochloric acid in the market is 37%. That means 12 molar concentration. In that case, we have to mix 8.3 ml of hydrochloric acid to make it 1 liter with distilled water to get this concentration of 0.1 normal. Here, replaceable hydrogen is 1. That means the molarity and the normality is same for this solution. Okay. Now let us see the experimental instrument. This is the SAHLI'S hemoglobinometer. It has the plastic body, which has the brown in color. Body color is black, fitted with white ground glass at the back side for good visual expression. It is the slit in which experimental tube can be introduced. Both sides there is a standard tube of brown prism. This is the experimental tube of same diameter. Both sides of it is etched the different measurement. This side is the gram, 2 to 24 gram, and this side is percentage, 20 to 160. This is the capillary tube for collecting blood. The glass made. At the back side it is the rubber tube, and this is the mouthpiece. Now, how do we function with this element? Extra to this element we require here, that is the stirrer, glass stirrer, and the dropper. So first of all, in the methodology, during experimental time, experimental tube will be cleaned and filled with hydrochloric acid up to 2 gram mark. Come to here. This is the experimental tube, and this should be cleaned. This should be washed, and then this hydrochloric acid is to be filled here carefully up to 2 gram mark. Right? Now, with the second step, micropipette, capillary micropipette should be filled with blood up to 20 ccm. This is the capillary tube. Blood is sucked in, and it will be sucked in up to this point, that is 0.02 ml. This blood will be transferred into it carefully. When it is transferred, the tip of it will touch here, and blowing inside, the blood will be pushed into the hydrochloric acid solution. Then it's carefully given out. After that, mix the blood with HCl glass rod and wait for 10 minutes. With the glass rod moving up and down, it is mixed continuously for 10 minutes, and it will help the proper dilution, proper hemolysis of the blood by means of hydrochloric acid. Okay. Again, after 10 minutes, again dilute the mixture, that is the hemolyzed blood, with the distilled water for proper color development. During this mixture, color has been changed due to hemolyze, and now water is added to it drop by drop with the dropper, and simultaneously it is mixed so that the volume of the content rise up. But not only that, the color changes from red to light brown. Color fades away. When this color comes approximately to the standard color here, then the mixing of drop of water will be stopped. Now, this tube should be placed into the comparator box. Comparator box is for the color matching, and when color is matched, that is brown color is matched with the predetermined standard color, then the value is taken out or recorded from these two graduations. This color, this number, the value obtained number is then compared with the values of standard values that I have mentioned before. But remember that this process of method is suited for only oxyhemoglobin. In case of methyl-hemoglobin, sulfur-hemoglobin, or even fetal hemoglobin, the result will be different due to hemoglobin concentration. Alright, so but I have mentioned a visit. You can visit this reference I have given to you for your proper visual experience if you like. And if you like my lectures, then you can subscribe to me and refer it to your friends. So thank you for this time. Meet you in the next class. Okay.